Carbon dating and DNA analysis are two powerful scientific methods used to determine the age of biological materials and analyze genetic information. These techniques play a crucial role in various fields, including archaeology, forensic science, evolutionary biology, and medical research. While carbon dating helps scientists determine the age of ancient artifacts and fossils, DNA analysis provides insights into genetic relationships, hereditary traits, and forensic identification. Carbon dating, also known as radiocarbon dating, is a method used to determine the age of organic materials by measuring the amount of radioactive carbon-14, C14, remaining in them. This technique was developed by Willard Libby in the late 1940s and has since become an essential tool in archaeology and paleontology. All living organisms absorb carbon from the atmosphere, including the radioactive isotope carbon-14. When an organism dies, it stops absorbing carbon, and the C14 present in its tissues begins to decay at a known rate, with a half-life of approximately 5,730 years. By measuring the remaining amount of C14 in a sample, scientists can estimate its age, up to about 50,000 years old. Carbon dating has been instrumental in determining the age of ancient artifacts, fossils, and historical structures. It has helped archaeologists date prehistoric human remains, identify the timeline of early civilizations, and confirm the authenticity of historical documents. However, carbon dating has limitations, including contamination risks, sample size constraints, and reduced accuracy for specimens older than 50,000 years. The process of carbon dating involves several key steps. Scientists first collect organic material such as bones, wood, charcoal, or shells for analysis. The sample is then cleaned to remove contaminants that may affect the accuracy of the results. After cleaning, the carbon content is isolated for measurement, using advanced techniques like accelerator mass spectrometry, AMS, to determine the remaining C14. The measured C14 is then compared to standard decay rates to calculate the approximate age of the sample. Despite its limitations, carbon dating remains a vital tool in understanding historical and prehistoric events, shedding light on the origins of human civilization and the Earth's past climate conditions. DNA analysis, also known as DNA profiling or genetic testing, is a scientific process used to examine genetic material to identify individuals, study hereditary traits, and detect genetic disorders. DNA, or deoxyribonucleic acid, carries the genetic instructions that determine an organism's characteristics. DNA analysis is widely used in forensic science, medical diagnostics, evolutionary studies, and ancestry research. It has revolutionized fields such as criminal investigations, where DNA evidence can link a suspect to a crime scene with high precision. Additionally, DNA analysis helps identify genetic mutations associated with inherited diseases, allowing for early diagnosis and personalized medical treatments. The DNA analysis process involves several steps to extract, amplify, and examine genetic material. First, DNA is collected from biological sources such as blood, saliva, hair, or skin cells. The DNA is then isolated from the collected sample using chemical or mechanical methods. Next, the polymerase chain reaction, PCR, is used to amplify, make copies of specific DNA segments, ensuring enough material for analysis. The amplified DNA is then separated and analyzed using gel electrophoresis or DNA sequencing techniques. Finally, the obtained DNA profile is compared to reference samples for identification or genetic study. Advances in DNA technology, such as next-generation sequencing, NGS, and CRISPR gene editing, have expanded the possibilities of DNA analysis. Scientists can now identify ancient human relatives, trace ancestry with high accuracy, and even explore potential treatments for genetic disorders. Both carbon dating and DNA analysis have numerous applications in science and society. In archaeology and anthropology, carbon dating determines the age of ancient human remains and artifacts, while DNA analysis helps trace human migration patterns and evolutionary history. In forensic science, DNA profiling is a crucial tool in solving crimes by identifying suspects and victims through genetic evidence. In medical research, DNA analysis is used to study genetic diseases, develop targeted therapies, and explore gene editing for medical advancements. In environmental science, carbon dating helps scientists study past climate changes by analyzing preserved organic materials such as tree rings and ice cores. Both techniques continue to evolve, contributing to breakthroughs in multiple scientific disciplines. While carbon dating provides a window into the past, DNA analysis unlocks the mysteries of life at a molecular level, shaping the future of medicine, law enforcement, and evolutionary research. Subscribe for more updates.